And the floor is yours.
Okay, great. Well, thank you everyone for tuning in. I'm really excited to be here today and talk to you about some of my dissertation work on color and visual system evolution in birds. I will say that most of this work, I'm now at the Natural History Museum. Most of this work was a collaboration between folks at UCLA, the Moore Lab at Occidental College, and the NHM too. And so I'm pretty sure I don't need to tell this crowd how incredibly colorful birds are. You can also see from this bird rainbow. I just love having it laid out because I think it really emphasizes just the amazing diversity of colors and patterns that birds display. And these colors really seem to span the entire rainbow. And before I dive too much into bird coloration specifically, I just wanted to give like a little primer on color itself. Um, so, what we call color is really just the reflection of visible light. And visible light is made up really of all the colors of the color spectrum or all the colors of the rainbow. And we really can't see all of those colors as light is traveling through the air. But something like a prism allows us to visualize each of those individual colors. So when light from a source, say either the sun or a light bulb or a flashlight, hits this prism, it's separated out so that we can see each of these individual colors. And this separation happens because light travels in different wavelengths of different sizes. And so, as light passes through the prism, it bends and slows this light and puts it out at different angles. So, we can actually see these different colors. So, here at the top, we see that red travels in really long wavelengths, indicated by the distance between the peaks here. Orange travels in slightly shorter wavelengths, and then violet here travels in the shortest wavelengths. And when we see color, really what we're seeing is the wavelengths of light that are being reflected off of the surface of an object. So here, for example, this red ball, it's absorbing all wavelengths of light except for red. And so this is why we see the red color reflecting off of that surface. And in a similar way, orange balls will do the same thing, absorb all the colors of light except for orange, and so on and so forth for all those colors. Black and white are slightly different. So the color black actually absorbs all wavelengths of light, and the color white reflects all wavelengths of light. And so the colors that we're seeing are really just a product of light being differentially reflected and absorbed by different surfaces. And so going back to the birds, all of these really diverse colors of birds has really fascinated biologists for well over a century. And because of this, we know quite a lot about the colors that birds produce and how they're producing each of these colors. And although birds also have really colorful eyes and skin, legs, and even eggs, today I'm specifically focusing on the colors that they produce in their feathers. And so there are two main ways that birds produce feather color. One of the ways is to produce color using pigments. They deposit pigments into their developing feathers, and pigments are just basically chemical compounds that selectively absorb and reflect different wavelengths of light. And so basically, you can think about paints. Paints use pigments to create their color. And for the same way, birds will use specific pigments and put them into their developing feathers. And so the main class of pigments in birds is called melanin pigments. These pigments are also found in our skin, our hair, and the fur of many other animals like dogs and cats. Birds use melanin pigments to make the colors black, gray, brown, rufous, and it also plays a really important role in patterning because it really is just the differential expression of melanin in the developing feather that creates these really striking patterns that we see, kind of the scaling or streaking patterns that we see on different birds like quail. And and one other thing about melanin is that it can actually be synthesized by the birds on their own, and it's pretty present. Present pretty ubiquitously across all birds. And melanin pigments themselves are a really structurally robust compound. And so when they're present in feathers, they can add a lot of structural integrity. So you may notice when you're looking at gulls, majority of their body is white, but those wings and the tips of the wings are dark. And this is because the deposition of melanin in those feathers actually really helps those feathers to resist abrasion and wearing, especially because they're so heavily used for flight. The structural integrity of melanin has also been shown to have some functions in parasite resistance, particularly against feather lice that feed on these feathers and cause early breakage. So the other main pigment class in birds is called carotenoids. And so carotenoid pigments are responsible for yellow, red, and orange in birds. And many plants produce carotenoids, but birds can't actually synthesize carotenoids on their own. So birds actually need to obtain carotenoids from their diet in order to put them into their developing feathers. So they can either take that carotenoid pigment directly from the plant product and put that color into their feathers, or they can chemically modify that carotenoid, but they can't produce the carotenoid on their own.
Whitney,
Yes.
just for 1 second, I did put a question in the chat. Feel free to enter your questions on the chat or after the program. Okay, back to you.
Okay, great. And so in order for birds to create these like very highly saturated carotenoid-based colors like house finches, birds have to consume a lot more carotenoids than are present in a normal diet. And so in house finches, the kind of brightness of that red is thought to be kind of an honest indicator of fitness because they're advertising that, I've been able to eat all of these carotenoid rich foods. I've been able to put that into my feathers and have a really healthy body condition. Another pigment in birds is called psittacofulvin, and this pigment is actually specific to parrots. And they have their own special color or pigment to make the colors red, yellow, and orange. So instead of carotenoids, they're using this psittacofulvin. Another unique kind of set of pigments that are only found in one bird family are the tarasin and turacoverdin, and these are found only in turacos. And so tarasin makes red in turacos, and turacoverdin makes green. And one interesting thing about turacos is that they actually Turacoverdin and tarasin are copper-based pigments, and because turacos live in Central Africa, it's one of the kind of richest copper belts. It's possible that they are obtaining a lot of copper into their systems because of their diet, and a high copper accumulation can be really damaging to birds. And so it's possible that these unique pigments evolved specifically to be able to detoxify high levels of copper in their feathers. But definitely more work needs to be done on these two kind of special pigments. And as far as we know, turacoverdin is actually the only green pigment that we know to be found in bird feathers. So another way that birds make feather or make color is through what we call feather structure. So we can think about a bubble. It mostly looks clear. It doesn't really have a specific color, but when light reflects off of that bubble at different angles, we can see kind of a whole rainbow of colors. And that's kind of how structural coloration works. So structural coloration is really in birds is the result of varying the composition of pigments and proteins in the feather nanostructure. And in this way, the light interacts with the cellular structure of the feather and the underlying pigments to reflect different colors. And so blue is an example of structural coloration. To our knowledge, we don't know of any blue pigments that are found in bird feathers. And so any blue that we see in birds is produced by structural coloration. So for example, this indigo bunting, this is a cross section of the feather, and we see that there is kind of this layering of keratin, spongy keratin, air matrix, and then black melanin on the bottom. And so when light hits that feather, it absorbs all wavelengths of light except for blue, and we see the blue color on this indigo bunting. If you were to put that feather in water and pulled it out and it was all wet, it would actually look either black or gray because the water is is changing the interaction of the light hitting that feather. And then once you dry that feather again, it turns blue because it kind of brings those microstructures or nanostructures back, and the light can interact with it in the correct way. Um, Structural coloration is also responsible for iridescent colors like those in hummingbirds. So here we can see kind of a different feather microstructure where we have a kind of more organized system of keratin and melanin rodules, keratin again, and more rodules. And this makes it so that when the light hits this microstructure, it reflects different colors depending on the viewing angle. And so I'm sure you all are aware, like when you see a hummingbird from some angles or when it's in bright sunlight, it turns towards you and you can see that gorget full on in like full color, like Anna's hummingbirds in the magenta throat. But if it's in the shade or if it's looking away from you, that throat gorget patch can look completely black. And so these are just two examples of how feather microstructure can produce really different colors. And structural coloration also makes purple feathers, it makes glossy or shiny feathers, and it also can make some kind of special black and special white colors. So, kind of your normal black or like pigmented black that you might see on like a crow, for instance, that's just pigment that's in those feathers. And then if you have just a regular white, those are just unpigmented feathers. Um, that would be kind of your regular white. But birds can also make this structural black that's called super black, and it actually absorbs more wavelengths of light than just your normal pigmented black. And we see this in things like this bird of paradise. We see it in Ramphichelys tanagers. And these birds are often having these kind of really black, super black feathers next to very bright patches of color. So it's thought that it's a way to be able to enhance those bright colors. And then for structural white, we see this in birds like rock ptarmigan. We see it in the brilliant white caps of manakins. And I actually have a colleague who's a postdoc at the NHM with me who's studying structural white because it hasn't been really studied before. Lastly, I just wanted to talk about green in birds because this uses kind of a mixing of mechanisms, kind of like how we mix two different colors of paint to make green. Birds also mix two different mechanisms to make green. And so they're using yellow carotenoid pigment and blue structural feather structure to make green. And so here is this budgie. It's a really common pet bird. Breeders have bred this bird to be different colors. So this is kind of your one of the budgies that has been bred to be green, but breeders have also bred these birds to be blue. And so this blue budgie was bred to not be able to have to synthesize the yellow carotenoids. And this other bird, this other budgie, has been bred to only be able to have the yellow carotenoids, and it's been bred not to be able to make those blue microstructures. And so we know that birds can make all of these different colors. But what I was really interested in for my dissertation work was really how do we characterize or quantify this color diversity in birds? And because I have this background of working in museum collections, I was really interested in taking an image of a specimen, cropping out all of the parts that aren't feathers, so taking out the cotton eyes, the legs, and the tags, and then being able to output the specific color categories that the bird is making and also count how many different color categories the bird's making. And so to do this, we came up with this R package called Charisma. And so R is just a computer programming language that's really widely used across many different fields, but especially in the biological sciences. And one thing that I'll mention about museum specimens is that it's possible to get kind of artifacts of color when you're imaging museum specimens because of the way the feathers overlap. So you can kind of see here in this, let's see if I can highlight. This area here looks like kind of brown or gray, and that's just because of the way that the feathers are overlapping each other. And so if we were to just kind of naively put this bird into a program and try to output the colors, we would get these patches here where we're getting kind of an artificial brown when it would probably need to be black. And so what we did was implement a way to be able to smooth out those color patches to get rid of those artificial kind of color patches and smooth it out so that we're not getting like an overrepresentation of gray or brown in our data sets. And so our outputs from Charisma look something like this. We get a smooth version of our original image, and then we get our distinct color categories for each of our bird specimens. We also get the total number of color categories, so the K value here. So this bird has 5 different colors. And then we also get the proportion of the image that is made up of each of our color categories. And so we ran Charisma for 30 or about 30 tanagers to both validate that Charisma was making the correct color calls and also to be able to model color evolution across this group. And so to validate that our Charisma classifications were doing a good job, we compared our Charisma color calls to colors that were identified by an expert in tanager coloration. So this table here is going to populate with numbers for hits, which are when the expert and Charisma both called the color or said that the color was present. The misses are when the expert says the color is present, but Charisma didn't call it. Correct rejections are when both the expert and Charisma didn't call the color. And then false alarms are when Charisma called the color, but the expert did not. And so here we'll want to see high numbers for hits and correct rejections and low numbers for misses and false alarms. And we do indeed see that we have almost 100% hits and correct rejections for our expert and Charisma color calls. And so this is telling us that our Charisma program is working pretty well. And so to investigate color evolution in these tanagers, we mapped our Charisma color calls on a phylogenetic tree of our tanagers. So this phylogenetic tree basically shows each of these tips is a different species. Birds that are more closely related to each other are closer on the tree. So here, if I can get my highlighter again, if you look at these 2 birds here, they're each other's closest relatives. You can see that these branches are pretty short and they're pretty closely related. And because those branches are short, that means that that split between those 2 species happened not too long ago. If you go along this branch and make it to this other species, we can see that this is the sister species of these 2. And so basically birds that are farther away in branch lengths are more distantly related to each other until we get here. And this would be the most recent common ancestor of all of the bird species in our tanager tree. And so using Charisma, we were able to populate this tree and show where all of these colors occur on all of these different tanagers. And we are also able to estimate what colors the most recent common ancestor of this tanager might have. And from our analyses, we estimate that it probably has black, brown, yellow, green, and blue. And then what we can do is map kind of where these colors have been gained or lost across our phylogenetic tree. And so here is an example of brown. We see that it's been lost in this lineage here, but then later it's been gained again here. And then we can also see that there's a gain of it in these lineages here. And we can do that for multiple colors. So we see some losses of blue and a gain of blue. We see some losses of green and a gain of green. And we see some losses of yellow and a gain of white also. So it's kind of cool to be able to see, you know, where have these colors been gained or lost? What might those kind of past tanagers looked like? We can also look at how fast colors are evolving in tanagers. And so we find that some colors have equal rates of gains and losses. So if you look at these arrows, the arrows up are showing the gain rates of brown, and the arrows down is showing the loss rates. And so these colors, white, brown, and yellow, are gaining and losing these colors at an equal rate. And brown seems to be doing this a lot faster than white and yellow. We also have some colors that have different rates for gains and losses. So green is losing, it is being lost faster than it's being gained, and blue is being gained faster than it's being lost. And the remaining colors on this color chart have pretty slow rates of evolution compared to these rates of evolution. And so here we were just looking at the human visible colors that birds make. And earlier I talked a lot about the human visible colors make, and I talk about visible light, but there's more to light than just what we can see. And so birds are actually capable of producing colors in the ultraviolet spectrum. And these are usually made by structural coloration. And so we can't see the UV signatures with our eyes, but we can see some evidence of UV coloration both under blacklight and under using UV photography. And we also know that birds can actually see UV colors as well. And we know this from studies of bird vision and also bird behavior. And we know that the ability to see UV isn't limited to just birds. And in fact, many fish, amphibians, reptiles, and even some mammals have UV vision as well. And although the vertebrate visual process is really highly conserved, we do know that the pathway to UV vision differs in non-avian vertebrates, and this points towards an independent origin of UV vision in birds. So just a kind of quick primer on bird vision. This is a diagram of an avian eye, and you may notice that it actually looks pretty similar to our eyes. So like us, bird eyes use a cornea and a lens to kind of filter and focus light to the back of the eye, which is known as the retina. And then within the retina, there are two different types of photoreceptor cells. So there are photoreceptors called rods and photoreceptors called cones. The rod cells are mostly for detecting kind of dim light, and they function in motion sensing and peripheral vision and night vision. The cone cells are what's important for bright light and important for color vision. And here you'll notice that I have cone cells in 4 different colors in this diagram, and that's because birds have 4 different kinds of single cone types in their eyes, and each of those cone types is sensitive to a different wavelength of light. And so here, these peaks are each of the sensitivities for each of those 4 different cone types. And so in birds, those cone types are loosely associated with red, green, blue, and ultraviolet. And when we compare bird vision to human vision, we see that birds actually have an expanded color vision. And this is because we only have 3 cone types, single cone types in our eyes. And those peak sensitivities are associated with red, green, and blue. That bird vision has their peak sensitivities are kind of more evenly distributed than ours. Those green and red peaks are pretty close to each other, and that's why there are people who are kind of red-green colorblind when those peaks overlap. And so here again, I'm showing you the peak sensitivities of each of those cones. And just like us humans in birds, these cones have an ability to shift around a little bit in their peak sensitivities. And actually, specifically for the UV cone, it can shift from inside the UV to outside of the UV. And so when that peak sensitivity of that UV cone is within the UV range, we call that a high ultraviolet sensitive visual system. If it's outside of that range, we call it a low ultraviolet sensitive visual system. And from here on out, I'll try to refer to those as high UVS and low UVS. And so from prior research of genomic sequencing and looking at this opsin called the short wavelength sensitive opsin, or the SWS1 opsin, we know that we can actually infer the sensitivity of the visual system by just looking at the genomic sequence of that SWS1 opsin. And being able to really infer this visual system sensitivity based on a single sequence is a really powerful tool to be able to investigate the evolution of visual system and color in birds kind of across the entire bird tree of life. And so having these tools really allows us to think more about this question and why are birds so colorful? And there are a variety of hypotheses for why kind of color and visual system have evolved in birds, and I'll go over some of them in the next few slides. So we know that plumage color can be really important in both sexual and social signaling. Birds use their color to either stand out or show off and to be able to attract mates. And UV color has been shown to play a really important role in mate choice. And UV reflecting plumage in particular can allow for signaling between conspecifics while excluding predators who may not be able to see or may not have UV vision. And so if we find a correlation between birds that make UV plumage and have a high UVS visual system, then it's reasonable to assume that signaling is involved. Light environment or habitat may also play an important role in color and visual system evolution. Plumage coloration can be really useful for camouflage. And while a lot of birds camouflage by being the color brown to blend in with things like tree trunks or dirt and leaf litter or rocks, another way that birds can camouflage is to be really brightly colored. So this red-crowned parrot here, you might not think that it's able to really camouflage itself, but we know that they forage really high up in these really leafy trees. And I don't know about you, but I've been out there looking at parrots and they'll fly into a tree and they will completely disappear. So birds don't just have to be brown to be camouflaged. It just really depends on what their background is and what they're trying to camouflage with. We also know that many leaf surfaces and fruits reflect UV. And so in addition to the varying amount of sunlight that penetrates through the forest canopy, this makes a really kind of complex visual environment. So there's a lot of birds that live in like low light forest habitats where having a high UVS visual system might help birds to navigate through those complex environments better. Foraging may be another thing driving the evolution of color and visual system in birds. So we know that many fruits have a waxy surface that reflects UV. And so birds with a high UVS visual system may be able to find fruit in these kind of complex visual environments. We also know that some small rodents leave urine tracks that reflect UV, so it could be helpful for some predators to have a high UVS visual system as well. Lastly, one thing that we know birds must balance with having a high UVS visual system is the risk of photo damage to their retina. So we know UV rays can cause damage to our retinas, and that's the same in birds. Our eyes do a pretty good job of filtering out UV before it hits our retina, but we still have to, be careful and not look directly at the sun and wear sunglasses. And so birds, especially those that are really long-lived, and live in certain habitats like open habitats, aquatic habitats, they're at really particular risk for photo damage. And so there has to be some way to be able to balance out the benefits of a high UVS visual system and the risk of photo damage. And so in order to really kind of tease apart all of these hypotheses, we first have to know the evolutionary history of this visual system sensitivity across the bird tree of life. And so here I'm presenting this kind of radial phylogenetic tree of my results. So again, this tree is just representing how closely related birds are to each other. So each tip here is a different species. I have just over 400 species on this tree, and it spans pretty much the entire kind of diversity of birds. And so here I can kind of identify some of the main groups here. We have our non-passerine birds, starting here and going in this direction. And it's a diverse group of birds, including things like ducks and pigeons and parrots and shorebirds. We have the start of our passerine birds here. This smaller group is a group we call subausine passerines, and these are birds like flycatchers, pittas, wood creepers, and then we have all of our songbirds that starts here and goes in that direction all the way to kind of the end of our phylogeny here. And songbirds make up just over 50% of all bird species. Um, and they include birds like warblers and cardinals and tanagers and scrub jays and all those fun species. And so the tip colors here are the actual visual system sensitivities. The red tips are low UVS visual system. The blue tips are the high UVS visual system. And then the branch colors are the estimations of what those kind of ancestral bird species visual system was. So here at the center of our tree, we see that there's an estimation of a low UVS visual system for the most recent common ancestor of birds. And this makes sense to us when we think about an independent evolution of ultraviolet sensitive visual system in birds because it evolved independently from other vertebrates. And so the ancestral birds started with this low ultraviolet sensitive visual system, and we see that a high UVS visual system has evolved multiple times across this tree. And so here in these different colors, I'm just highlighting some of the different lineages where we see high UVS evolving. In terms of non-passerine birds, we see it evolving in sand grouse, gulls, trogons, motmots, parrots. In the sub-Aucine passerines, we only see it in one group, the antbirds. In songbirds, we see it in quite a few different groups. We see it in lyrebirds, fairy wrens, long runners, some vireos. And then once we get to this group called the passerides within the songbirds, these are birds like wide-eyes, warblers, tanagers, robins. And we see that it's pretty ubiquitously high UVS visual system. And so in the next few slides, I'll go over some of these results in a little bit more detail. And so in terms of low UVS visual system, we found this to be the case in birds that are known for to be high aerial flyers. So this included birds like raptors, like hawks, eagles, and falcons, and then also in swifts. And so as I talked about before, UV vision, there is a risk of photo damage from UV hitting the retina. And so it's been hypothesized that these high aerial predators are at an increased risk of photo damage. And previous studies have actually shown that raptors have lenses that are pretty good at filtering out UV. And so there's a pretty low UV transmittance to the retina. And so maybe retaining this low UVS visual system along with the low UV transmittance to the retina would make sense in terms of avoiding damage to the retinas. We also found another group of birds that had a low UVS visual system, according to our SWS opsin, and that's the hummingbirds, which was actually kind of surprising. It was a little bit difficult to obtain the full SWS1 sequences from hummingbirds. And from the partial sequences that we did find, we found that it had a low UVS sequence. But we know from behavioral studies that hummingbirds can actually distinguish UV colors, and they're actually, in fact, very good at it. And we also know that they're nested in a clade with birds that have low UVS visual systems and also nocturnal species, things like oilbirds and nightjars. And so there are a few possible reasons that we might be getting this low UVS signal from hummingbirds. One is that they might be missing the SWS1 opsin or it's highly modified. So maybe at some point they actually lost this SWS1 opsin and after that, they might have even had a unique pathway to UV vision. And what's really interesting is that hummingbirds have another sensory system where they have evolved a unique pathway, and that is the pathway for their sweet sensing. So that is another gene that they or kind of molecular pathway that they evolved uniquely. And so I know that people in a lab at Princeton are actually studying this pathway in hummingbirds. So we'll see. Well, I'm excited to see what happens with that research. Excuse me. Next, we find that gulls and skimmers actually have a high UVS visual system. And so, oh, excuse me, let me just take a breath. And so yeah, this was actually a little bit surprising also. We know that water reflects a lot of UV, and the shore and kind of aquatic habitats that they live in would seemingly expose them to a lot of UV, especially with the UV reflecting off the water, and it would increase their risk of photo damage. And so we were thinking, well, we know that fish exhibit UV coloration, and so maybe having a high UVS is helping them to find prey. But we know that high UVS visual system is actually really rare in shorebirds and other seabirds, and many of these birds are also catching fish. So we know that a high UVS visual system isn't necessary to locate fish. And so another alternative is that, these gulls and skimmers are kind of more coastal and kind of terrestrial coastal birds. And so further investigation into kind of gull vision and foraging behavior is needed to understand why they might have this kind of high UVS visual system, or why they may have evolved this. And so within the songbird diversification, there is an infraorder that I mentioned called the passerides. And within this group, high UVS is present in all of those species. And it could have several functions in this group. So many of these species live in kind of closed forest habitats that are known to have low light, known to be pretty complex visual environments. So having a high UVS visual system could, one, help them with foraging on the berries and other fruits that have that waxy UV reflecting film. It could help them navigate through this environment better. It could also help them with signaling. So we know that many birds in this group are quite colorful. Many of them also have UV reflecting patches. So this could help them communicate in those low light, closed forest habitats. It could also help them with cryptic signaling, where, their patches are visible to their conspecifics, but not to their predators. We also found high UVS visual system in parrots. And so, one thing about parrots is that we do know that they are very long lived birds. So, they do have a high risk of photo damage. Um, but as I mentioned before, parrots have a special pigment called psittacofulvin. And this psittacofulvin is found in birds, and it makes in parrots, and it makes the red, orange, and yellow in their plumage. And we also know that carotenoids are known to be effective antioxidants and immunostimulants, and that they are actually present in parrots. So it's not that carotenoids are not found in parrots, it's just that they're not found in the feathers. They're actually found in the oil droplets of the retina. So more work needs to be done on this, but it's possible that the evolution of psittacofulvins in parrots allowed for this high UVS visual system, and for them to be able to save their carotenoids for the oil droplets in their eye to reduce their photo damage to the retinas. And so, in summary, we found that a low UVS visual system was likely the ancestral visual system in in all birds, and that a high UVS visual system has evolved up to 18 times across the bird tree of life. We also found some kind of expected results, like a low UVS visual system in raptors, a high UVS visual system in passerides, and also some intriguing results that kind of warrant further investigation, like a low UVS visual system in hummingbirds and high UVS systems in gulls and parrots. And so now we know that we can characterize both color and look at patterns of color evolution in birds, and we understand how the visual system sensitivity is evolving across this bird tree of life. And so now what I'm working on is another aspect of bird coloration, and kind of taking into account all the colors that birds are making across their entire bodies. And so if you're familiar with this paradise tanager, then you might know that it actually has a really bright red rump, and that's completely obscured in the side view of this specimen. And we also know that this paradise tanager reflects a lot of UV in its plumage. And so, in order to be able to capture the full coloration of birds, we're making 3D digital models of these bird specimens. And so what we do is set a specimen up on a turntable. We sync our camera with the turntable so it takes a picture with every rotation, and we do that from 3 different angles. And then we can stitch together all of those photos to produce these really high resolution models that work really well for being able to capture all the colors that are present across the entire bird. It's also really great for accessibility, so we're able to upload these specimens online. We include a scale bar with them, and so you can take really detailed measurements. If folks aren't able to come to the museum to actually measure the birds, they can do it digitally and still do their research, which is great. And so we can make these models both in human visible spectrum on the left here and in UV spectrum on the right. And by doing this, we can quantify all of the colors that the bird's making. We'll be able to run these models through Charisma to figure out the proportions of each of these colors and which colors they're producing. And then we'll be able to look for correlations between plumage coloration, their visual system sensitivity, and then the types of habitats that birds are living in to figure out and investigate why birds have evolved such incredible coloration. And so with that, I'd like to thank you all so much for your attention. And I have a lot of collaborators and funding sources to thank. And yeah, feel free to reach out to me via email with questions or ask them now.